Okay, so we're still in a basic science mode. We haven't quite got to anatomy and physiology yet. But at least now we're in a living organism, the cell. The study of cells is known as cytology, and one of the most important things that has developed for biology over the last 200 or 300 years is the cell theory. The cell theory states that all organisms are composed of cells and cell products, that the cell is the simplest structural and function unit of life, that an organism's structures and functions are all due to the activities of cells, and that cells come only from previously existing cells. There are at least 200 types of cells in the human body with various shapes, sizes, and functions. Organs, tissues often refer to the shape of the cells as squamous, cuboidal, columnar, polygonal, stellate, spheroid, discoidal, fusiform, fibrous. These polygonal, stellate, spheroid, and ovoid and discoid you're not going to see very often. The most common ones we'll see are squamous, which are thin, flat, and scaly looking when looked at from above. Cuboidal, which are roughly the same width as height. Columnar, that are taller than they are wide. A fusiform shape we're going to see in smooth muscle. And fibrous cells would make up a lot of skeletal muscle as well as other places. So you can view a cell in different types of sections, longitudinal or cross, and so your shape is going to look a little different depending on whether you're looking in longitudinal or cross section. Most human cells are about 10 to 15 micrometers in diameter. A micrometer is really, really small, as you might guess. It's 1/1,000,000th of a meter, 1,000th of a millimeter. There are exceptions. Egg cells are very large. They can be up to 100 micrometers in diameter, and egg cells in some organisms are visible without the microscope. Some nerve cells can be over a meter long. They won't be very wide, but they're very long. And there are limits on the size of cell. When you have an overly large cell, it won't be able to support itself. And a lot of that has to do with the geometry. Increasing diameter, you increase the volume faster than the surface area. So the volume is going to be proportional to a cube of the diameter, while the surface area is proportional to a square of a diameter. The end result is you're trying to do too much and you don't have enough ways to get materials in and waste out. Microscopes have improved greatly. We started with a simple light microscope, which allowed the discovery of the cell. They then became up to compound light microscopes, and we were able to see the plasma membrane, the nucleus, and the cytoplasm. We now have transmission electron microscopes that give us the ability to see details. They're going to use beams of electrons rather than visible light. And scanning electron microscopes also can be used to produce a dramatic 3D image at high magnification and resolution, but only on the surface features. So the trade-off for using the transmission or the scanning electron microscope is you have to use a stained specimen, and it has to be a fixed specimen. The major components of a cell are the plasma membrane, which defines the cell boundary. It's made up of proteins and lipids. Its composition will vary between cells, and it will also vary between regions of the cell. The cytoplasm is within. Inside the cytoplasm are the things we talk about, but we can't see with a light microscope. Organelles, cytoskeleton, inclusion bodies if they're present. And it's all surrounded by that clear gel we call cytosol, or intercellular fluid. The extracellular fluid is outside the cells, so any fluid outside of a cell, including tissues, is extracellular fluid. If it's within a tissue, it's also referred to as interstitial fluid. So interstitial fluid is a type of extracellular fluid. And then we have blood plasma, lymph, cerebral spinal fluid, in addition as extracellular fluid. So here is a representative cell with all kinds of things labeled on it, and we will get into that momentarily. So we start with the outermost region in the plasma membrane that defines the boundary of the cell. It's a pair of dark parallel lines when viewed with an electron microscope. And those dark parallel lines are the phospholipid bilayer that sets up. It has an intracellular face and an extracellular face. Most of the membrane is lipids, mostly phospholipids. So the phospholipids are 75% of the membrane lipids. If you remember from last week, they are amphipathic molecules. They arrange themselves in a bilayer. Their hydrophilic phosphate heads will face the water outside the cell and the water environment inside the cell, while the hydrophobic tails are going to be directed towards the center of that membrane, and that serves as a water barrier. They do drift laterally, move around. They're not static. Most of them are not static in place. In addition to the phospholipids, there are cholesterol molecules. Cholesterol molecules are very important parts of cell membranes. Not all cholesterol is bad. They make up about 20% of the membrane, and they help to hold the phospholipids still and stiffen it. They make the plasma membrane a better water barrier, but at the same time they make it a little more fluid so that phospholipids and proteins can drift laterally. Glycolipids make up about 5%. A glycolipid is a lipid with a carbohydrate attached to it. They're phospholipids with short carbohydrate chains on the extracellular surface, and they will contribute to the glycocalyx, which is a carbohydrate coating on the surface of the cell. So if we take an artist's rendering of the cell, plasma membrane, we find all kinds of nice little molecules. We've got our phospholipid bilayer. We've got proteins embedded into the membrane, shown in purple. And we've got cholesterol molecules. Those membrane proteins are about 2% of the molecules, but about 50% of the weight of the membrane. If they are passing completely through the membrane, they are known as transmembrane proteins. They will have a hydrophilic region in contact with the cytoplasm or the extracellular fluid on each side of the membrane, and a hydrophobic region that keeps them within the lipid portion of the membrane. Most of them are glycoproteins. They have carbohydrates attached to them. Some of those do drift around. Others are anchored to the cytoskeleton. There are peripheral proteins in many cases that adhere to only one side of the membrane, either the inner or the outer. Then interface, they're usually tethered to a transmembrane protein and the cytoskeleton. So here are transmembrane proteins passing through the membrane. Receptors are going to bind chemical signals and trigger internal changes. They can cause the production of a second messenger within the cell receiving the chemical message, and we'll talk about second messenger systems shortly. Many of them are enzymes. They will catalyze reactions, which could include the digestion of molecules or the production of second messengers. There are also channel proteins that allow hydrophilic solutes and water to pass through the membrane. There are some that are always open. They are called leak channels, and some that are closed sometimes, called gated channels, and they only open when they're triggered, and the things that can trigger them are different. So if we have a ligand-gated channel, it will respond to the binding of a chemical messenger, a ligand. We have voltage-gated channels that will respond to changes in electrical charge. And we also have mechanically gated channels that will respond to physical stress on the cell. Some proteins are carriers. They will bind a solute and transfer it across the membrane. If that carrier requires ATP to do its function, it would be known as a pump. Many of the glycoproteins on the outside of the surface are cell identity markers. Each one of us has signs on our cells identifying that cell as belonging to us, so that our immune system can distinguish between us and something invading us. A lot of membrane proteins are also cell adhesion molecules, or CAMs. They mechanically link cells to one another and to the extracellular matrix. So those second messenger systems are very good at amplifying responses within the cell. They're called second messenger systems because the first message comes from outside the cell, and then something happens inside the cell to amplify that response. So, for example, the stimulation of a cell by epinephrine. The first messenger would be the epinephrine binding to an epinephrine receptor. That surface receptor on the internal portion is going to activate what is known as a G protein. A G protein is an intracellular peripheral protein that, instead of using ATP for energy, uses GTP for energy, or guanosine triphosphate. A G protein relays a signal to adenylate cyclase, which then converts ATP to cyclic AMP. And this cyclic AMP becomes the second messenger. The cyclic AMP will activate all kinds of kinases. Kinases are enzymes that add phosphates to other enzymes. Sometimes when you add a phosphate, you turn on something. Sometimes when you add a phosphate, you turn it off. And this becomes important because a lot of drugs use G proteins as second messengers. So first message is epinephrine binding, activates a G protein. The G protein activates cyclic AMP, and cyclic AMP activates cyclic AMP kinases. And again, the the advantage of a second messenger system is you get an amplification. One epinephrine can activate lots of different kinases. So I mentioned the glycocalyx, that is the carbohydrate moieties of glycoproteins and glycolipids along the external plasma membrane. They are unique in everyone except for identical twins. Their functions are numerous. They provide protection. They help with the immune system. They help defend against cancer. Transplant compatibility involves the glycocalyx, cell adhesion, fertilization, and embryonic development. On the surface of the membrane, we can find some extensions, including microvilli, which are 1 to 2 microns of the membrane that may serve to increase the surface area. So they can provide 15 to 40 times more surface area to cells that have them. So they're in places where we are absorbing things. Some absorptive cells are very dense. They appear as a fringe called the brush border, which we'll see when we talk about the digestive system. Some microvilli contain actin filaments that are tugged toward the center of the cell to milk the absorbed contents into the cell. Here are microvilli in cross-section and transverse section. Cilia are longer. They are hair-like processes that can be 7 to 10 microns long. They are primarily motile for moving things across the surface of a membrane. Multiple non-motile cilia are found on sensory cells of the nose, but motile cilia are less widespread. We do find them in the respiratory tract, the uterine tubes, ventricles of the brain, and ducts of the testes. There could be between 50 and 200 on each cell, and what they do is beat in a waving motion that sweeps liquid material, across the surface of these cells in one direction, caused by a power stroke followed by a recovery stroke. So cilia move within saline layer at the cell surface, and mucus floats on top of that. So the cilia help move the mucus from the lower parts of the respiratory system out to the upper parts of the respiratory system. There is a core of motile cilium consisting of microtubules called the axoneme. It has a 9+2 structure, 2 central microtubules surrounded by a ring of 9 pairs. The ring of 9 pairs anchors the cilium to the cell as part of the basal body. And then there are dynein arms that crawl up the adjacent microtubule, causing the cilium to bend. This requires the use of ATP to provide an energy source. So the green here are cilia, and they are beating in a way that moves material in one direction. Here is the cross section. And so here's what we get. We move mucus, for example, up the respiratory tract until we get rid of it, and then that individual cilia gives you the power stroke and the recovery stroke. Cystic fibrosis is a hereditary disease, a relatively common genetic disorder in which cells make chloride pumps but fail to install them in the plasma membrane. The chloride pumps don't create enough. Without the chloride pumps, you don't make enough saline layer on the outside cell surface, and the thick mucus is going to plug the respiratory tract and pancreatic ducts, giving you inadequate digestion of nutrients and poor absorption of oxygen. Results with also have chronic respiratory infections. Mean life expectancy for someone with cystic fibrosis is now 44 years, which is quite an improvement over 30 years ago. Another extension is a flagellum, which is a whip-like structure. We only have one cell with a flagellum in humans, and it's only found in 50% of humans. It's the tail of a sperm. It has a whip-like structure with an axiome and identical to cilium, but it's much longer and stiffened by coarse fibers that support the tail. Movement is undulating, snake-like kind of corkscrew. There are no power stroke and recovery strokes. The membrane can also reach out, creating pseudopods, which are continually changing extensions of the cell. They're used for cellular locomotion. They're used for capturing particles, engulfing materials. So the plasma membrane and the organelle membranes are selectively permeable. They allow some things through, but prevent others from coming through. There are a couple of ways you can do that. There are some that are passive mechanisms, which require the use of no ATP. They're driven by the random motion of molecules going down concentration gradients. We can also use them for filtration, diffusion, and osmosis. Active mechanisms are going to require cellular energy, and cellular energy comes in the form of ATP. And there are also carrier-mediated mechanisms that use a membrane protein to transport substances across the membrane. So filtration, the particles are driven through a membrane by physical pressure. This wouldn't really be a way to move things into and out of a cell, but filtration is important. So if you're filtering water through small solute gaps between the cells, you get filtration pores in the capillaries. Simple diffusion is the movement of particles from a place of high concentration to low concentration due to constant spontaneous molecular motion. Molecules collide and bounce off each other. Substances diffuse down their concentration gradient, so they go from areas of high concentrations to an area of low concentration. Will happen in air, water. It doesn't require a membrane, but when it becomes important to us in cell transport, we will use a semi-permeable membrane, which is the plasma membrane. Things that will affect simple diffusion include temperature. Because it increases the temperature, you increase the motion of particles, you speed up the rate of diffusion. Larger molecules diffuse more slowly. The steepness of the concentration gradient will make a difference. The amount of surface area of the membrane will make a difference, as will the permeability of the membrane to the substance. If we happen to be talking about water moving through the plasma membrane, then we are talking about osmosis. Osmosis is the net flow of water through a selectively permeable membrane. Water, like other things, will move from an area of high concentration to an area of low concentration. So when you think about this, we really need to think about the amount of solute. So a high water concentration means a low amount of things dissolved in it, solute. And a low water concentration is because there's more solutes dissolved in it. There are water can go through a phospholipid bilayer, but osmosis is mostly enhanced by a group of proteins called aquaporins that are specialized for water passage. They can speed osmosis by installing more aquaporins, and osmolarity, osmosis is a crucial consideration when you're using ISB fluids, because osmotic imbalances underlie things like diarrhea, constipation, and edema. So osmotic pressure is the hydrostatic pressure required to stop osmosis, and increases as the amount of non-permeating solute rises. So non-permeating solutes cannot pass through the membrane. Proteins would be an example. Hydrostatic pressure is the fluid pressure on the membrane. Reverse osmosis is a process of applying a mechanical pressure to override osmotic pressure, and that's used to purify water. Osmolarity is the osmotic concentration, the quantity of non-permeable solutes per liter of solution. So body fluids contain a mixture of many chemicals, and osmolarity is the total osmotic concentration of all the solutes. Typically for blood plasma, tissue fluid, and intercellular fluid, it's about 300 milliosmoles per liter. Tonicity is the ability of a surrounding solution to affect fluid volume and pressure in the cell. That's going to depend on the concentration of solutes, those that will not cross the membrane. If the solute is more concentrated on one side of the membrane, it will cause osmosis to occur, which gives us three possible scenarios. A hypotonic solution will cause a cell to absorb water, swell, and sometimes burst, or lyse, as we call it. That solution will have a lower concentration of solutes than the intercellular fluid, distilled water, for example. A hypertonic solution will cause the cell to lose water and shrivel, which we call crenation, and it has a higher concentration of solutes than the intracellular fluid. In an isotonic solution, we don't get a change in cell volume because the concentration of solutes in the extracellular and intracellular fluid are the same, and that's why we use normal saline, 0.9% sodium chloride. We still get a movement of water back and forth, but there's no net change in the movement. So we've got your hypotonic, isotonic, isotonic, and hypertonic. If we use a carrier-mediated transport, we can move some things across the cell membrane. Carriers exhibit specificity for their particular solutes. A solute is the ligand that binds to the receptor on one side of the carrier protein, and then that solute will be released on the other side of the membrane unchanged. We can, because we are using a protein, we can saturate the system as soon as you get enough that you are using every single transporter. You've reached the transport maximum. You can't go any faster than that. Whoops. Three kinds of carrier proteins: uniports, symports, and antiports. Uniport carries a carrier that moves one type of solute, calcium pump, for example. A symport is a carrier that moves two or more solutes simultaneously in the same direction, so a co-transporter. Sodium glucose transporters are co-transport. And an antiport carrier is one that moves two or more substances, but each one goes in opposite directions, so a counter-transport. The sodium-potassium pump that we're going to see a lot of in the nervous system is an example of a counter-transport pump. There are three mechanisms of carrier-free mediated transport: facilitated diffusion, primary active transport, and secondary active transport. In facilitated diffusion, the carrier moves solutes down its concentration gradient, so it does not require any ATP. Solutes attach to a binding site on the carrier. The carrier changes conformation and releases solutes on the other side of the membrane. In primary active transport, you get a very similar situation, but the carrier protein needs to use ATP to move that substance and move it against its concentration gradient. Calcium pump uses ATP to get calcium out of the cell. Sodium-potassium antiport uses ATP when getting sodium and importing potassium. So here we have facilitated diffusion. Molecule enters the protein, binds to the protein changes shape, and it's released on the other side. Secondary active transport, carrier moves a solute through the membrane but only uses ATP indirectly. So sodium glucose transporters are an example of secondary active transport. Moves glucose into the cell up its concentration gradient while carrying sodium down its concentration gradient. So it depends on the primary transport being performed of the sodium-potassium pump, so it doesn't use ATP itself, but it does use the gradient created by a pump that uses ATP. So that's why it's secondary active transport. So in the sodium-potassium pump, or the NAK pump, each pump cycle consumes one ATP and exchanges 3 sodiums for 2 potassium, keeping the potassium concentration higher and the sodium concentration lower within the cell compared to the extracellular fluid. And it's going to maintain sodium gradients, which will allow for secondary active transport, and it will regulate solute concentrations and osmosis and cell volume. Vesicular transport is the movement of large particles, fluid droplets, or numerous molecules at once through the membrane in vesicles. A vesicle would be described as a bubble-like enclosure of the membrane. If it's bringing material into the cell, it's known as endocytosis. If it's taking material out of the cell, it's known as exocytosis. Endocytosis can have special names. We have phagocytosis, which is the engulfing and destroying of large particles, cell eating, if you will. The pseudopods surround the object, fuse to form an internal phagosome, and that phagosome will merge with lysosomes to give you a phagolysosome, and the lysosomes contain digestive enzymes that break down the materials inside the phagosome. Pinocytosis is taking droplets of extracellular fluid that contain molecules useful in the cell. So pinocytotic vesicles in the cytoplasm you can think of as cell drinking. And then there is receptor-mediated endocytosis, in which particles bind to a specific receptor on the plasma membrane. That structure forms called a pit, and the pit is covered with clathrin, and the clathrin-coated vesicle is directed to its destination inside the cell. Here are the receptor-mediated endocytosis. Transcytosis is the transport of material across the cell, capturing it on one side and releasing it on the other. We'll see that in the digestive system. Exocytosis is the discharge of material from the cell. So releasing insulin by endocrine cells, sperm cells release enzymes penetrating the egg, mammary glands release milk sugar. And it will also function to replace plasma membrane lost by endocytosis. Okay, cytosol, next layer. Cytosol is clear, viscous, watery colloid within the cell. Contains enzymes, other proteins, amino acids, ATP, electrolytes, dissolved gases, and metabolic waste. The cytoskeleton is a network of protein filaments and cylinders which support, determine the cell shape, and organize cell contents, directs the movement of materials within the cell, and contributes to the movement of the cell as a whole. That cytoskeleton is composed of microfilaments, intermediate filaments, and microtubules. And here we see the constituents of the cytoskeleton. So microfilaments are thin filaments about 6 nanometers thick made of actin. They form a terminal web or the membrane skeleton. The intermediate filaments are 8 to 10 nanometers thick. They're found within skin cells. They're made of keratin, and they give shape and also resist stress. Microtubules are 25 nanometers thick. They consist of protofilaments made of tubulin, which radiate from a centrosome. They come and go. They help to maintain the cell shape, hold organelles, act as railroad tracks or highways for motor proteins, make axionemes for cilia and flagella, and they will form the mitotic spindle during cell division. Organelles are the internal structures that are within the cytosol. They have membranes around them, the nucleus, the mitochondria, the lysosomes, the peroxisomes, endoplasmic reticulum, and Golgi apparatus are membrane-bound organelles. There are some organelles that are not membrane-bound. They don't have a membrane around them. Ribosome, centrosome, centrioles, and basal bodies. So the nucleus, the third thing we can see with a light microscope, is the largest organelle. It's usually about 5 microns in diameter. It contains cells' genetic material. Most cells have only one nucleus. There are a few cell or cell products that are anuclear. They do not have a nucleus. And there are a number of cell types that are multinuclear. The nucleus is surrounded by a double membrane called the nuclear envelope. In that envelope, there are pores called nuclear pores formed by a nuclear pore complex that regulate molecular traffic through the nucleus. So the nucleus becomes selectively permeable, similar to the plasma membrane. It's supported by a web of protein filaments called nuclear laminin. And within the nucleus, we would have nucleoplasm, which would include the DNA, chromatin most of the time, one or more nucleoli, which is where ribosomes are produced. Coming out, we have endoplasmic reticulum, which is a network of interconnected membranous channels called cisterns. Endoplasmic reticulum comes in two forms. Rough endoplasmic reticulum has parallel flattened sacs that are covered with ribosomes. It's involved in protein synthesis. Proteins will be built into rough endoplasmic reticulum, and inside the rough endoplasmic reticulum, the proteins will be modified and folded correctly. There is also smooth endoplasmic reticulum, which does not have ribosomes attached to it. It's not involved in handling of proteins, therefore, but it is used to synthesize things like steroids and other lipids to detoxify alcohol and other drugs, and it can be involved in calcium storage. Ribosomes are granules of protein and RNA that are used to build proteins. They can read the genetic code, the messenger RNA, and they provide a platform for the assembly of the amino acids into proteins as it's specified in each code. We find them in nucleoli, in the cytosol, and on the outer surface of rough endoplasmic reticulum. From the endoplasmic reticulum, most things will move to the Golgi complex, which is a system of membranous cisterns that synthesize carbohydrates and modify newly synthesized proteins. It's going to sort, splice, and add carbohydrate moieties to some, and then ultimately it will package them into membrane-bound Golgi vesicles. Those vesicles may become lysosomes. Some vesicles transport materials to the plasma membranes. Others become secretory vesicles that store the materials later. Lysosomes are a package of enzymes bound by a membrane, and they are intracellular hydrolytic digestion proteins. They can break down proteins, nucleic acids, complex carbohydrates, phospholipids, and other substances. They can be used for autophagy, which would be the digestion of surplus organelles within the cell, or they can trigger autolysis, which is the digestion of the cell itself, so-called cell suicide. Peroxisomes are similar to lysosomes, but their enzymes are different. Their function is to use molecular oxygen to oxidize organic molecules. Their reactions produce hydrogen peroxide. Catalase can then break down the excess peroxide to water and oxygen, neutralize free acids, detoxify alcohol and other drugs, and a variety of blood-borne toxins. They're also used to break down fatty acids for the use of the mitochondria. Although they are present in all cells, they are much more abundant in the liver and kidneys. Proteasomes are hollow cylindrical organelles that dispose of surplus proteins. So they have enzymes that break down targeted proteins into short peptides and amino acids for recycling in the cell. Mitochondria, are the powerhouse of the cell. They synthesize ATP. They change their shape, although we often look at them as a bean. Surrounded by a double membrane, the inner membrane has folds called cristae. The spaces between the cristae are the matrix, and the matrix contains ribosomes and enzymes used for ATP synthesis and small circular DNA called mitochondrial DNA. Again, powerhouse of the cell. Energy from glucose is transferred into the bonds of ATP. Centrioles are short cylindrical assembly of microtubules arranged in 9 groups of 3 microtubules each. 2 centrioles lie perpendicular to each other within the centrosome, a small clear area in the cell, and they play a role in cell division. And they also form basal bodies of the cilia and flagella. Inclusions are places where products are stored in the cytoplasm. They could be accumulated cell products like glycogen granules, pigments, or oil droplets. They could be foreign bodies, including viruses, intracellular bacteria, maybe even just dust particles. They never enclosed in a unit membrane. They are not essential for cell survival. Whoops, went the wrong way. And that brings us to the end of our tour of the cells. Thank you. I will be posting your work shortly.